Hello everybody, I'm Dr. Sean Gibbons here at the Institute for Systems Biology. To start off, I got to level with you. Germaphobes out there, you might find this a little queasy, inducing. I got to tell you, you're just covered in microbes. Every inch of your body is completely coated in microorganisms. There are trillions of microbial cells in and on our bodies. And for about every human cell, there's a microbial cell in our bodies. And that'll shift a little bit. You might be more microbe or human depending on when your last bowel movement was, but it's about on par. And today's discussion is going to be about this kind of novel organ of the human body that we've really only come to appreciate in the last couple of decades. My lab studies the ecology and evolution of these microbial communities and how they work, how they assemble, who interacts with whom, just try to understand the basic ground rules underlying the communities. My lab and many other labs around the world trying to understand this system. We employ both computational and experimental approaches to try to understand these systems, see how they work, and then ultimately, once we understand the rules of the system, we can begin to engineer it. We can begin to take it apart, put it back together again, and if it breaks, fix it. Well, okay, that's a big question. Biome is an ecological term, so biome is just all the flora and fauna that live in a particular environment. So you could think of a forest as a biome, all the animals and plants living in that forest, or the ocean is a biome. And then micro, obviously, is tiny. Microbiomes are just tiny environments of the organisms that live in those environments. And the human microbiome is just all the assemblage of all the microorganisms that live in and on our bodies. Everything from bacteria to fungi and viruses. So lots of tiny little organisms living in our guts, on our skin, in our armpits, and so on. Where do these organisms live? Well, the human body is a very diverse ecosystem. So there are many sublocations within our bodies. Our gut is the most jam-packed, full of microbes. So most of the microbes in our bodies live in our gastrointestinal system. And it's a very specific environment, right? It's dark, it's anaerobic, which means there's no oxygen around, it's moist, it's warm, and that's very different from, say, the barren wasteland of our skin, very dry, exposed to the sun, and the types of organisms that can live in these different environments vary drastically. So if you looked at the sort of ecological similarity between a gut community and a skin community, you can kind of quantify this distance in species shared between the systems. You get numbers that are similar to, how similar is a desert from a rainforest, right? Really, really, really different ecosystems, and they're only separated by a few feet or a few inches on our bodies. And this microbial ecosystem in and on our bodies, we really can't avoid having it around. Large bodied creatures like ourselves, animals, plants, during the course of our evolution, which is a very small snapshot of the total time life has been on Earth, we've had to evolve in this milieu of microorganisms. We're just surrounded by bacteria and other microbes, and we either had to learn how to beat them or how to join them. We're either these walking happy meals that they eat, or we somehow find a way to coexist and live together. And the human microbiome is for the most part a story of sort of neutral or positive relationship between us and our microbiota. We've learned to live with them. We're actually a pretty stable, nice environment to live on for a century or so, if we're lucky. And so there's a bit of a détente where we don't kill each other, we actually kind of provide conditions conducive to the success of both groups of organisms. So we can think of our bodies as not being completely human, but actually being an assemblage, a chimera of different species interacting to produce an overall being or organism. And so a lot of the health and well-being of our body system is outsourced and pent up in our microorganisms that have been with us for so long and been co-evolving with us. So we provide a home for them, we provide food to them, and for us, they provide protection from pathogens, they help us digest our food, they help educate our immune systems, and so much more. this is actually the subject of a grad student's work that I worked with in Erica Holmes' lab at MIT, Claire Duvalet. She did a meta-analysis, which is an analysis across many different studies of just asking the question, what does a sick microbiome look like? You know, when it breaks down, what breaks? And the answer to the question is it's complicated. It's complicated to define what a healthy microbiome is. It seems to be that we have different ecological niches within our bodies that need to be filled in order for the functioning of our bodies to be complete. But there are many different species or organisms that can plug into those niches. So any given one of us can have sampled from a universe of microbial diversity, a subset of that diversity that is functional or that is healthy, and it's not necessarily overlapping with the person next to us. So there's many ways to build a healthy microbiome. So that's the kind of long-winded answer to what a healthy microbiome is. We don't really know yet, but there's lots of ways to construct one. What we can see from certain diseases is particular ways that the microbiome can break. So in a disease like, this is again from Claire Duvalet's work, a disease like colorectal cancer, we can see that there are certain bugs, certain bad bugs potentially, that seem to be enriched in patients that have disease. So that's the major signal of that disease, is you see an overabundance of particular taxa that tend to not be there or to be at very low abundance in healthy people. So enrichment for bad bugs. There are other diseases, however, where you have the opposite effect, where you get a depletion of the good bugs. So inflammatory bowel disease is one of these examples, like Crohn's or ulcerative colitis, where what we see is a depletion of a whole guild or group of microbes that is responsible for a very important ecological function, which is the production of these things called short-chain fatty acids. And for example, butyrate is one of these fatty acids that's produced. And it's been shown that this butyrate molecule in the gut actually helps our immune system to dampen its response and to be less inflamed. And if we lose those organisms that provide that function to us, then this disease can erupt. And so you can either have situations where a pathogen comes into your system and that makes you sick, or you can have situations where the ecology of your gut is degraded because you lack certain good bacteria. So there's many ways for things to go wrong in the microbiome, and there's many ways for things to go right in the microbiome. And we are at the very forefront still of really understanding those rules. The short answer is yes, you could. So for example, you could go get food poisoning, and that would very quickly change your microbiome. Maybe not permanently, but it would definitely change your microbiome. But I guess the real question is, can we predictably change the microbiome to, say, improve our health, for example? And this is a hard problem. So many of you may have heard of things like probiotics, where you try to introduce an organism into your gut microbiome. Well, a lot of commercial probiotics that you can get at the grocery store, these are organisms like bifidobacteria, these taxa that tend to be very low abundance in our guts, or at least in the guts of adults. And often if you sequence the poop of someone who's taking these probiotics, you find that they'll take the probiotic and you'll see that organism in their stool, but as soon as they stop taking the probiotic, the organism washes out. It goes away, it doesn't stick. So it's hard to actually put a new species into the gut microbiome and make it land and make it stick permanently. Sometimes you only need a transient change in your microbiome to have some sort of health effect. So we know that even dead bacteria can sometimes modulate our immune response. So if you cooked microbes and fed them to someone, you could have a reduction of inflammation just because the molecules in the outer membranes of these dead bacteria are actually having a temporary drug-like effect on that person's system. But if you wanted to actually change and engineer and reshape the microbiome, we still are at the really early stages of being able to do that. And essentially, there's no clear way to engineer the system. It's like a game of pickup sticks, and you change one little thing, and there's repercussions and ripple effects to the rest of the system. And so what we really need in order to do this well is an understanding of the niche structure and the ecological interactions that are occurring within a given person's gut microbiome. All those metabolic interactions of one microbe feeding another, the host giving, the human giving molecules to a microbe. once we understand all those interactions, we can begin to find deficits in a given person. So say I sequence your microbiome, I look at your microbiome, and I find, oh, you're missing species X, Y, and Z, or functional capacity X, Y, and Z, and then maybe I might have a freezer full of particular probiotics to make a customized treatment for you that fits your ecology. That's sort of a sci-fi version of the future for what we might be doing, but currently we don't yet know the basic ground rules for how to do this. So the answer is yes. We can change our microbiomes. I think eating fiber is usually a good thing. Having a complex, diverse diet that includes a lot of plant material kind of promotes diversity and promotes health in the gut. But as far as steering the ship in a particular direction or changing in a particular way to achieve a certain health outcome, we're still far away from actually being able to do that. So Purell, some of you may know, is this hand sanitizer. It's essentially alcohol in a gel form, and you rub it on your hands to clean your hands after using the bathroom or what have you. It is a little known fact that people who overuse Purell, that really use a whole lot of Purell, way too much, most people would never use this much, end up getting plantar's warts and get other problems on their hands because they've actually stripped away so much of their own microbiota and their own immune system from their hands that they are more susceptible to viral infections. And I would argue that in modern society, we are actually depleting our microbial diversity too much. The vast majority of microorganisms in the world are either neutral to our health or beneficial to our health, and it's only a very small subset of bad actors that are pathogens that actually cause us harm. And it's having an intact and functional ecosystem on our hands that can act as a barrier to those organisms being able to invade. So it's well known in the gut, for example, that for like Clostridium difficile infections tend to be enriched in people who take antibiotics, for example. They're left vulnerable to these infections because they depleted the natural ecosystem, the indigenous microbiota that would otherwise have prevented that invasion. So I think products like Purell, these scorched earth policies of germophobia, just get rid of all the bacteria, I think in the end that's probably a net negative to our health. We shouldn't be doing that. Most studies looking at transmission of disease in public spaces like public restrooms show that basic use of soap and water, non-antibacterial soap, just soap and water, is completely sufficient to prevent the spread of influenza or other viruses. And actually the spread of bacterial pathogens is pretty rare, at least in developed countries. It's only in situations where there's an outbreak of a particular pathogen, like a cholera outbreak, where you'd even have to worry about that kind of bacterial transmission of a pathogen in a public setting. So the overuse of these sterilization agents might actually be a net detriment to our health, and I actually would suggest that people not use these products. First off, antibiotics are like a miracle of modern medicine. They've saved hundreds of millions, if not billions of lives over the last century. And it used to be a long time ago that you got a paper cut and that got infected, that could be the end of your life, right? And we don't live in that world anymore. And it's a better world to live in to not be dying of infectious disease constantly. So antibiotics are great. They're a godsend, they're wonderful. However, there's been a cost to antibiotics that has been underappreciated over the past century or so that we're just now beginning to understand. One of those costs we've known for a long time, which is the rise of antimicrobial resistance. So genes that are resistant to antibiotics, so I should back up a bit, antibiotics that we use in medicine, they already existed in the natural world for millions and millions of years. These are chemicals that microbes use for warfare against one another. And so genes that resist these antibiotics already exist in the soil or in the water or what have you. And so when we start using high doses of antibiotics in humans, those pathogens that are infecting humans will eventually find their way to one of these soil bacteria, for example, that might contain one of these antimicrobial resistance genes for that antibiotic and transfer it to the pathogen. bacteria are notoriously promiscuous when it comes to transferring DNA across each other. You know, on our level, it would be unthinkable for a banana tree and a zebra to mate. But in the microbial world, organisms that are equivalently distant can actually exchange genetic material. And so with the use of antibiotics, we've seen the influx of these pre-existing resistance genes out in the world, or through evolution, the evolution of these resistant organisms. That's the first cost. The next big cost that we didn't really think about very much in the beginning was a depletion of our indigenous microbiota. So antibiotics are a scorched earth policy. Broad spectrum antibiotics, they're not just hurting the bad bug that's infecting you, but they're also killing off a lot of the native microbes that are in and on your body. And this can leave you even more vulnerable after having taken them to other types of infections or secondary infections and so on. And over long spans of time, over generations, we actually see a slow and steady loss of microbial diversity, some of which is potentially passed from mother to child, and a venting of this diversity and a loss of the ecology of our bodies. And in the Western world, we have seen an uptick, mild uptick, in certain autoimmune disorders, overactive immune system type disorders that seem to be associated with use of antibiotics, changes in our diet, like eating less fiber, for example, and a steady loss of microbial diversity in our bodies. So I think the answer is, don't be afraid to use antibiotics. If you need antibiotics, please use antibiotics. You shouldn't not use antibiotics. If you have a viral infection, don't use antibiotics, of course. You need to be responsible about your use of antibiotics. But what we need to think about for the next generation of treating people with these drugs is, how do we recolonize or recover a healthy microbiome following antibiotic treatment? And there are a few ways to think about this. I know there are groups working on these pills you can swallow that have a capsule on the outside that only dissolves in the lower colon that contain activated carbon. And this can kind of soak up the antibiotic in the colon and actually prevent it from killing off your gut microbiota. So that's a way of sort of resisting the killing of microbes of an antibiotic treatment. And then you can also have a recovery side where there are these things called autologous fecal transplants, and we'll probably get to that later on in our conversation, which in that autologous just means from yourself. So I could take my poop and I could put it in the freezer when I'm healthy and store it for a rainy day. And then down the road, I get antibiotic treatment, and at the end of that treatment, I could take a poop pill of my own poop and recolonize my gut with my healthy microbes from before, as a way to sort of bring back that diversity after the treatment. So I think the future of medicine will incorporate these other treatments that actually help us to maintain and regenerate the microbial diversity that we initially had, even after these antibiotic treatments. Yes, it is as gross as it sounds, because it is literally what it sounds like. So if you haven't heard of fecal transplants, it is a strange concept to wrap your head around. Essentially, what it is you take a turd or a poop from a healthy person, you mix it up with some liquid, and you inject it into a sick person to try to make them better. So why would you do this? Does this actually work? This sounds kind of crazy. Well, it did sound kind of crazy until a few years ago. In my postdoctoral lab, my postdoc advisor, Eric Alm, has pioneered some of this work and started a company that actually creates material for these fecal transplants called Microbiome. These transplants have been shown to be incredibly effective for treating a very specific disease, which is C. difficile, Clostridium difficile infections. So I only have statistics from a few years back, but I remember a few years back, about 30,000 Americans every year were dying from Clostridium difficile infection. It is the second most common, I believe, hospital-acquired infection, second only to staph infections. It tends to strike in really young people or in elderly people who already have sort of compromised immune systems or compromised guts. And a big risk factor for getting C. diff is taking antibiotics. So you go in, you take antibiotics, you might get colonized, and the next time you have another round of antibiotics or another depletion of your diversity, suddenly you get sick and C. diff takes over and you get really bad diarrhea. 70% of people that go in and get treated with vancomycin antibiotic to treat this C. diff infection. 70% of people are fine, they get better, they go home. But about 30% of those folks, they end up getting what's called recurrent C. difficile. And once C. diff recurs, the likelihood that antibiotics are going to be able to get it in the next round or to cure it or resolve it is very, very low. And so recurrent C. diff, you end up getting in this vicious cycle of constantly having to be on different rounds of antibiotics. And some people live in this cycle for years to the point where their colons are so damaged they have to be surgically removed. Parts of them have to be surgically removed. And then that's where all these complications like sepsis can come in, and that's where people die, essentially. So it was, in some cases, an incurable disease, antibiotic resistant, really virulent, really nasty, killed a lot of people. But we figured out that if you just squirt a little healthy poop up someone's bum who has this infection, 95% resolution to that disease. There's been many clinical trials now. I think the efficacy varies between the high 80s and low 90s, but more than 90% of the time someone gets a fecal transplant, they're completely cured of this incurable disease that was killing people before. So it's really the first example of using the microbiome, engineering the ecology of the microbiome to treat disease. And I find this to be incredibly exciting. It shows that our field is relevant to medicine. And I would predict that in a decade or so, we might actually even see in medical schools entire programs where students will focus on the microbiome as one of their specialties. So I think the future is bright for using the microbiome to treat disease. Fecal transplants, this first example, they're trying to apply it to other diseases now. So I know it's being applied to inflammatory bowel disease. There has been more complicated. It's not quite as effective. And we seem to find this effective super poopers, where there are particular donors who are really good at treating patients with inflammatory bowel disease and other donors who have no effect. And so we still don't really know what the secret sauce is. We don't really know the active ingredient yet. And that's a lot of companies and a lot of labs are really quickly trying to pin that down, because there's a lot of pharmaceutical money involved in making products that can do this without having to give someone a poop transplant. But it's exciting. It's an exciting time to be in the field, because with this initial proof of concept, now we're starting to get more focused in creating therapeutics that target very specific diseases and just be able to engineer the microbiome to improve human health. A question I can't really answer at this point. There's really good evidence in a mouse model that the microbiome can induce weight gain. So there was experiments done probably 10 years ago at this point, where they took fecal material from an obese person and from a lean person, implanted that into mice, different mice, and found that on the same diet, mice that got the obese microbiome got fat, and mice that got the lean microbiome stayed lean. However, this has been really hard to translate to humans. So the signal in humans is much noisier. We don't see as clear of an association. And so I think it's a little premature to say whether or not your microbiome can make you fat. But there is evidence that it plays a role. It definitely plays a role in our metabolism, in what metabolites show up in our bloodstream after we eat a meal, in how we process food and how we regulate that process. And I'll give just one example of a paper I know about where they had a population of people, and they sequenced their microbiomes. They found this population of people have a very distinct type of community where they're dominated by an organism called Prevotella. And this other group is dominated by an organism called Bacteroides, a genus of a different species within that genus. And these are two kind of subtypes that these organisms don't coexist in the same person, and so they partition the population into these two groups. And if they fed a high fiber diet to these two different populations of people, they found that in the Prevotella microbiome group, people lost weight. But in the Bacteroides microbiome group, there was no weight loss. And this was a small cohort study, but there's been multiple papers now showing that for the same diet, you can have very different metabolic responses based on your microbiome. So I think the future is bright for being able to disentangle that and understand it better, and maybe use the microbiome as a tool to help modulate weight gain or loss. But we're a ways away from being able to do that. Yes, simple answer. There are literally hundreds of diseases that have been associated with changes in the microbiome. It's been really hard to tell whether or not those associations are causal or whether they're just happening, they're just sort of hitching a ride on whatever else is happening in your body, right? So is the microbiome different because the environment in your body is different when you're sick? Or was there a change in the microbiome that somehow precipitated a disease state? This is very hard to tell for most things. For Clostridium difficile infections, we know the causal role there. But for a lot of other diseases, it's harder and harder to say. Starting with cancer, there's a few examples where, actually recent examples, very exciting examples, where there does appear to be a connection between the microbiome and cancer. So with colorectal cancer, for example, it does seem that there are certain organisms that are normally very rare in the gut, like Fusobacterium is one of them, and a few others. I think they're often present in the mouth, but they're very rare in the gut. But they seem to be able to colonize polyps in the intestine. So oftentimes when you go for a colonoscopy, those of you who are a little older who go for colonoscopies, sometimes they'll snip the polyps. These are sort of precancerous little protrusions that they get rid of just to be safe. But these polyps are microenvironments that allow for the colonization of something like a Fusobacterium, which settles down and then forms a biofilm on that polyp. And at least in a mouse model, they've shown that these biofilms, these microbial biofilms, actually produce metabolites that can feed into oncogenesis or the induction of cancer. So in mice, at the very least, we have shown that colorectal cancer does have a causal link to this Fusobacterium colonization. It's been messier in humans. It's not as clear in humans that is the case, but I would guess there is some sort of feedback role between those changes in the microbiome and colorectal cancer. Other than that, there's a huge connection between the microbiome and the immune system. We know that there's feedback in both directions, from the immune system to the microbiome and from the microbiome to the immune system, and recent ways of treating cancer have really begun to rely upon the immune system. So treatments for cancer that use the immune system to attack the cancer, it has been shown that differences in people's microbiomes actually affect the outcomes of those treatments. So these treatments are more effective in some patients that have certain types of microbes in their guts. And a very recent paper, this was in mice, so again it's not humans, but in mice they found an 11 species consortium of bacteria that they could grow and feed into another mouse. They found that when they applied these cancer immunotherapies to these mice, they found the efficacy of the immunotherapy with this 11 bug cocktail was improved significantly. So perhaps the future of cancer treatment might go hand in hand with engineering the microbiome and the immune system to kind of work in concert to attack and resolve the cancer. So that's for cancer, but I could spend all day talking about the hundreds of other diseases in which the microbiome has been implicated. We know that a lot of what we eat, a lot of the complex plant molecules in our diets, we don't have the genes to break those down. We can't break down fiber ourselves. Even like the polysaccharides in breast milk, for example, when we're babies. A lot of those polysaccharides that mom puts in the breast milk, we can't eat them. They're specifically food for our microbes. But the breakdown products of some of those polysaccharides actually make their way into our bloodstream. So we can measure the metabolism of our microbiome in our blood metabolome. And we're just starting to kind of decouple and understand how different types of microbiota modulate this blood metabolic signature for each of us for a given diet. Yes, so this is another one you got to be careful to talk about. So, of course, formula feeding is perfectly fine and a great way to feed your kids. And oftentimes mothers need to use formula. There are lots of reasons why formula has to be the way to go. But there has been a lot of research recently showing that a lot of the components of breast milk, of our mother's milk, actually foster certain components of our microbiome, and that may have an impact on our health later in life. So one thing I just spoke about was the oligosaccharides that are present in breast milk. So a lot of the calories in breast milk can't actually be consumed by us. They're specifically consumed by particular bacteria, and so mom is controlling what bacteria can live in our guts. So mom is doing her own ecosystem engineering on the infant microbiome and making sure that the right types of organisms are in that kid's system to promote that kid's health and well-being. And some of those probably exist in formula, but the exact cocktail of them that really produces the right combination has been honed over evolutionary time very carefully. So the likelihood that formula is able to completely recapitulate that is not perfect. In addition to that, we know that the maternal milk contains a lot of antibodies, which are little molecules produced by our immune systems that are involved in binding things like bacteria. And recent work from a colleague at Fred Hutch, Megan Koch, shout out to Megan, has shown that if you don't provide maternal antibodies along with maternal microbes, so if a little kid is getting bacteria into their system but they're not getting those antibodies, you can actually mount an inflammatory response to your own gut microbes. So your immune system, when you're really young, has a really hard time distinguishing between self and non-self. And so there's all these complex organs and processes involved with making sure your immune cells don't attack your own tissues. And this education is constantly happening. But in addition to our own tissues, we also have to learn we don't want to attack the food we're eating. We don't want to constantly be attacking all the bacteria that are living in our gut. If we did that, we'd be constantly burning all of our resources and having inflammation in all these places we don't want it. And so part of what mom helps us do when we're young is train our immune systems, quiet our immune systems down to not overreact to our microbiome and treat our microbiome as self. And this can break down a bit when we don't have access to these antibodies. And so, there are human studies now showing that various factors early in life, like cesarean versus natural birth, or breastfeeding versus non-breastfeeding, when we're cesarean born or when we're not breastfed, our likelihood of later in life having an overreactive immune system, which could mean allergies, asthma, rheumatoid arthritis, what have you, are slightly increased. Now it's just a slight increase in risk. It's not like you're doomed to have a bad immune system if you're cesarean born. That's just not true. Most cesarean born kids are completely healthy and completely fine, but we do see that there is a slight increase in the risk for those things. And this is true across the developed world. If you look at Europe, the US, Australia, all these countries that have modern healthcare systems, modern lifestyles, between using Purell, using antibiotics, cesarean birth, no breast milk, there are all these multitudes of factors that are depleting our microbial diversity, and all of that seems to be feeding into the rise of these inflammatory conditions. This is called the hygiene hypothesis in my field, that these modern plagues of the immune system are actually caused by a disruption of our microbiota through these myriad factors. So where do I see the microbiome going in 5 to 10 years? Well, really, we've only known about it for the last 10 or 15 years, right? I mean, we've known the microbiome has existed for a long time. Van Leeuwenhoek, a couple hundred years ago, this Dutch guy who invented the microscope, scraped his teeth and looked under the lens and saw there was all these little creepy crawly things. And for a century or two following that, we were kind of limited to just that. We're just looking under the microscope. We can see that there's stuff there. Maybe we can grow it on plates like Louis Pasteur. But we didn't, most of the stuff that was there, we can't grow. It's called the Great Plate Count Anomaly. So when you looked under the microscope, about 90% of the cells you could see, if you tried to grow them on a plate, only 1 to 10% of them would actually grow on your media. And so we were missing a vast amount of this diversity that we didn't know was really there. So with modern sequencing technology, we were able to peer into the microbiome and see it for the first time, 2 decades ago, decade and a half ago. And that's really the birth of the microbiome field. So we're just in the very beginning, right? This is like Galileo invents the telescope, and we're just getting a first glimpse of this new world. So the last decade has been discovery. We've just been, it's like Darwin on the Beagle. It's like going to South America, walking around, seeing what's there, writing in our notebooks, classifying the diversity that exists. And that's kind of the hard slog that we've done for the last decade. What I envision for the next 5 to 10 years is really cashing in on all of that work. We've built this body of knowledge, we've built a platform for ourselves to really now go and do targeted work to really understand the rules underlying the system, which will then allow us to engineer and manipulate the system. And the ultimate vision of the field over the next decade will be to take what we've learned and actually make measurable impacts in the lives of ordinary people. We want to save lives and cure disease through the microbiome. And I think we've already seen a lot of that happen, and I'm nothing but optimistic that it's going to be the same going forward. So it's going to be an exciting time to pay attention to the microbiome field. So, watch this space.